properly stain it. So to begin with, it's very important that you have clean glass slides. So they should be scrubbed under tap water with some kind of cleanser, such as Ajax, on both sides of the slides. Even if the slides look clean, there's an oily residue. So be liberal with your cleanser on both sides of the slide. Rinse it thoroughly. Dry it with a brown paper towel or a Chem wipe. Do 3 or 4 slides, because although this technique looks simple, you don't always get the proper quantity of blood, and so every smear attempt is not successful. Lay those slides out in front of you long ways. In other words, directed away from your body once you have them clean. Some of the slides will be the slides to which you'll add the blood. One of the slides will be used as a slide to drag your blood across the slide, so that one will never actually be used as a smear. Then in addition to that, you're going to need a sterile alcohol pad, 70% isopropyl alcohol, and some type of blood lancet. You'll notice on the blood lancet it indicates which end to open it. That's so that you do not come in contact with the sterile tip or point of the lancet. So open it on the end, indicate it. Professor Sloan will demonstrate one method for properly lancing a fingertip. You want to disinfect your patient's finger with the alcohol pad. Lance on the lateral surface of the finger pad. They suggest that you lance with the barb going parallel to your finger ridges so it's not as painful. The main thing is definitely do not stick the patient in the nail beds, because that would be extremely painful. Once you're lanced, wipe off the first droplet of blood, and then the second and subsequent drops of blood can be applied to your clean glass slides. You want to make any comments about the lancing procedure?
Well, we would like for the patient to be seated, so again, if they feel faint, or in fact do faint, they don't have so far to fall.
You also should have gloves ready so that the moment that the procedure is over and you've collected your blood on the slide, you can cover your wound and you will not be exposed to contamination. So while the disinfected finger is drying, that is a good time to open up your lancet. And he will pull back the skin using his thumbnail. He'll pull back the skin on my finger to tighten the skin so that when he does prick me, it will in fact cut the skin. For those of us who have very tough skin, don't wear gloves when we work in the yard and so forth. Wipe off the first droplet with the alcohol pad. Now, sometimes for students that are just attempting this procedure, at that point it's easy to roll the finger over so that the blood hangs down from the finger. Okay. Once he's collected the blood, you can press the alcohol pad against the finger until it quits bleeding, then put your glove on. Pull the slide using as the slider back. Pause, allow the blood to run out left and right, then push the blood away from you. Don't do the other one since that first one's so good, because when she comes in, then they can see the size of the blood droplet. We will not save any of our blood materials, in other words, lancets, alcohol pads, any slides that you don't end up staining will all be disposed of in some kind of safekeeper, sharp container, but not in the trash cans where they can puncture the trash bags and expose the cleanup crew to body fluids. At this point, let's home in and look at this prepared blood smear a little closer. This is an example of an ideal blood smear in that the quantity of blood was just enough to cover about half the slide, and you ran out of blood before you got to the end of the slide. The usable portion of the slide is down here where you ran out of blood, and you want to see it feather out like that. You want to see the blood feather out on the end. Now this slide is not ready to stain until all the blood on that slide appears dull in color. At that point, the blood is properly affixed to the slide and it will not wash off in the staining procedure. But as long as you see any glossiness, then the blood would wash off if you began the staining procedure. So wait until it has that dull appearance. I saved this slide to show you about an approximate amount of blood that would create a good blood smear. If you put less than this, you're not going to get a nice distribution of blood on the slide. If you put more than that, you're not going to run out of blood. You're going to have blood all the way to the end of the slide, and therefore the red blood cells will be too close together, and you won't have a good field of view when you get to the microscope. So you hope to run out of blood and achieve these feathered ends like you see on this slide. Let's stop for a minute. So students allowed their blood smear to air dry until all of the blood on the entire slide surface was dull. Now remember, we only made one human blood smear, but you would probably have made 3 or 4. So you went from slide to slide with your lance fingers, putting a drop of blood on 3 or 4 slides. You come immediately along behind that with the glass slide that you used as your slider and smear that blood out, going from one slide to the next slide to the third slide without pausing much in between. If you do not get organized and move from slide to slide methodically, that droplet of blood that you put on that cold glass slide will coagulate, and then the blood will not smear out left and right across the slide as it should have, and so it won't be distributed properly down the length of the slide. So you do have to be organized, move along, and hopefully one of your 3 slides will be a successful blood smear with the feathered out ends. So you examine your slides and you pick the one that you like the appearance of the best to start a staining procedure. If you go to the sinks have a staining pan. In the staining pan are 2 metal slide racks, and you lay your slide across those metal slide racks. That way, when you flood the slide with stain, the stain goes down into the stain pan and is eliminated in a diluted form down the sink. We will be using Wright's blood stain. Wright's blood stain is a mixture of several stains, although it appears to you as dark purple or dark blue when you take it out of the container. In the Wright's blood stain, there is a red-orange dye called eosin and also a violet-purple stain called crystal violet. And different cells in your blood will have an attraction for either the red stain, the eosin, or the violet stain, the crystal violet. And so it will allow you to tell the difference between some of the various types of leukocytes or white blood cells on your blood smear. So that's the purpose of staining, is to reveal the differences in your populations of leukocytes or white blood cells. When you use the dropper bottle, be liberal with the stain. In other words, flood the entire surface that has blood on it thoroughly. Your lab procedure suggests a 10-second staining procedure. There are many different procedures, but we're going to attempt the 10-second one. So you can watch the second hand of the clock on the wall, and when 10 seconds is up, your procedure suggests that you then add tap water directly to the stain. You are to add about as much tap water as you did stain, and you are to blow to keep this stain and tap water mixture in contact with your blood smear. So the blow is a method of stirring the tap water and the stain together on the slide. You are to continue that blowing procedure for about 1.5 minutes. So you could have done several slides at once, or several students could be working on a single slide at one time. So you could have your slide, somebody else could have their slide, and a third person could have their slide. There's enough room around here that you can share the Wright's blood stain plus the squeeze bottle of tap water that several of you can get in here and work together. If for some reason your mixture washes off the slide, then add a little more water to the slide so that it does not go dry in that 1.5 minutes. At the end of a minute and a half, you are to dump the excess stain and water mixture off the slide. Then thoroughly rinse with the squeeze bottle all of the excess stain off the slide until no more Wright's blood stain is bleeding off of your blood smear. At that point, you are thoroughly finished with the staining procedure. And you'll notice I'm rinsing both surfaces of the slide because oftentimes when it's laying in the stain rack, the stain will crawl up under the bottom surface of the slide. So you must constantly keep in mind which side of the slide your human blood is on. Otherwise, you'll be working on the wrong side of the slide. Shake the excess water off the slide. And now you want to allow the slide to air dry. You can hurry this procedure along by blotting your blood smear in bibulous paper. You don't tear the paper out of the book. You just lay your slide gently in between the pages of the bibulous paper. Press down very gently. If you press hard, you're going to pop your slide. You're going to have to start all over again. But then once you press, that blots the excess moisture off the slide, and it will air dry much more quickly. You absolutely may not put a stained slide on your microscope until it is thoroughly dry, because if any of the water with stain residue gets on the lenses of your microscope, it will destroy those lenses. So we want it to be thoroughly dry before you go to the microscope. Besides that, slides do not move properly on the stage platform of your microscope until they're thoroughly dry. Otherwise, they stick or suction themselves to your stage and they don't move where you want to. But at any rate, once this is dry, we'll examine it with the microscope. That is the end of your staining procedure, unless you had another slide that you wanted to stain. So students, we have now made a human blood smear and stained it with the Wright's blood staining procedure. However, I do need to make one clarification. In the Wright's blood staining procedure, you use the Wright's blood stain, then added to it distilled water. Throughout the procedure, I said tap water previously, but it is always distilled water. So you add the distilled water to the Wright stain, blow it to stir it around, or rock the staining pan to stir it around. Then, after a minute and a half, you dump the excess off and thoroughly rinse the entire slide with distilled water. Blot it dry in your bibulous tablet, then let it completely air dry. At that point, you go to the microscope. Your objective under the microscope is first to see if you can identify the various kinds of formed elements. The formed elements in the field of view that you'll see under the microscope include red blood cells, which by far are the most numerous. Here's an enlarged view of an erythrocyte, a red blood cell. And you notice they don't have a nucleus. Then the other cellular components are leukocytes, which are your white blood cells. Leukocytes all will have a nucleus, but their nucleus comes in various shapes. Some have many lobes in the nucleus, and so they look very oddly stained, but the nucleus will be some shade of violet. At any rate, any of these violet nucleated cells are your white blood cells or leukocytes. And then there are some fragments of cells, which we call platelets, or in clinical terms we call thrombocytes, and those help you with blood clotting. So first, can you identify the difference between erythrocytes, red blood cells, leukocytes, white blood cells that have a nucleus, and platelets, the fragments that are many times smaller than the red blood cells? Once you can achieve that, what you then want to do is home in on some of the leukocytes, the white blood cells, and see if you can tell the neutrophils, which have sort of a light pink or lavender cytoplasm, from the eosinophils, which have distinct visible orange-red granules in the cytoplasm, or some shade of red granule in the cytoplasm, from basophils, which have violet, dark purple granules in the cytoplasm, from lymphocytes, which have no granules and are the smallest white blood cells, from monocytes, which have no visible granules in their cytoplasm and are the largest white blood cells. So we want to see if you can recognize neutrophils, eosinophils, basophils, lymphocytes, and monocytes. So that is your goal for your slide work once you get to the microscope. And in order to do that, you'll need to be eventually up on the yellow high dry power of your microscope.
Dr. Schlichter, and this is my name right here. I noticed from looking at the roster that there are some folks, I don't know if it's you or what, there's some folks in here with some difficult to pronounce last names. Well, believe me, you know, I know how you feel. I'm trying to grow up with that. But anyway. All right. So, well, before we do this, let me ask you something from this video. There is more than one way to stain blood cells. But there is one way that is the most common of all. One type of stain, which is by far the most common for staining blood. What was that? Which one? Wright's stain. Wright's stain. So these slides over here that you're going to be looking at are mostly done with Wright's stain, but you'll notice that some of them are done with Giem's stain. And you know, you can see the same stuff, but most of the time it's Wright's stain. Now you saw in this video that some cells take a certain stain a certain way, other cells stain a different way, right? They don't all take the stain the same way. Now, what's the purpose of staining it anyway? What if I took just a drop of blood like you saw there, and you took the other slide and you smeared it out there, and you let it dry, and you put that under the microscope? Are you going to tell very much from that? Not really. I mean, you could probably maybe tell the difference in sizes of cells, because most of the cells are going to be the same size, red cells, and there are going to be some that are bigger than red cells, but you know, there are different types of white blood cells. How are you going to tell one from another? Well, they have different staining qualities. So, you know, we're going to find out that there are 5 different types of white blood cells. How come they're not always all the same? I mean, how come, you know, the red cells look all pretty much the same, don't they? How come all the white cells don't look the same?
They do different things?
They do different things. They got different functions. They help us in different ways. So, because of that, you know, their internal structures are going to take up stain in different ways. That's how we tell what we know. All right. Now, when you get your slides under the microscope, you're going to see something roughly that looks like this. All right, so this is a picture of a peripheral blood smear on a slide. All right, so all of these cells in here are red blood cells, right? And these ones that have the different staining qualities are white blood cells, aren't they? And these little things up here are platelets. Now, why do we say in the composition of blood that the composition of blood is plasma and formed elements? Why do we say it's plasma and cells?
Because platelets are not.
Because platelets are not cells. Platelets are cytoplasmic fragments of a larger cell in the bone marrow. These cytoplasmic fragments break off from the larger cell and enter the bloodstream. So platelets don't have nuclei. Of course, mature red blood cells don't either. But platelets are fragments of a cell, not an entire cell. So that's why we can't say that the composition of blood is plasma plus cells. So technically, we have to say something that includes the platelets. All right. So now, not all of these red blood cells look the same, do they? Some look kind of solid, like this, and others look like they have a hole in the middle, right?
Mm-hmm.
And the reason for that is what? What's the shape of a red blood cell? Biconcave disc. It's plump around the edges and thinner in the middle. This allows it to bend and flex and get into places smaller than itself. Right? So that's why some of these look like little pink Cheerios. All right. Now, this particular slide here shows you 3 of the 5 types of white blood cells. So here are neutrophils, here's a lymphocyte, and here's a monocyte. Now, remember in the Wright's stain, it's made up of eosin and crystal violet. Those are the 2 main components of the stain. Certain white cells will take up the eosin or acidic part of that stain very well, such as eosinophils, which are not on this slide. I'll show you in a minute. Other white blood cells take up the basic component of Wright's stain, or the crystal violet part, and I'll show you that in the basophils. Right? These particular cells right here, the neutrophils, actually take up both the eosin and the crystal violet, sort of equally. So if you take a reddish orange and a crystal violet and mix them together, what color do you get? Something in between, right? More like a lavender color. And so for the most part, when you look at these slides, when you come to a neutrophil, it's going to have a mixing of the eosin and crystal violet. Their cytoplasm is going to have more of a homogeneous lavender color. All right, so that's what you see here. Thus the name neutrophil, neutral. There's something between eosin and crystal violet, neutrophil. The last part of that name, phil, P-H-I-L, comes from the Greek verb phileo, meaning love. It's one of the Greek words for love. So neutrophils love both the eosin and the crystal violet. So you get a neutral thing. All right? The other thing about neutrophils is that they have a lobed nucleus. In other words, the nucleus is not one solid round or semi-round thing. It has lobes, anywhere from 3 to 6 lobes. That's characteristic of neutrophils. So when you look at your slides, the cell that you're the white blood cell that you're most commonly see is the neutrophils, because they are the most numerous. They're by far the most numerous of the white blood cells. So when you look at your cells, you're going to be seeing a lot of neutrophils, anywhere from 55 to up to 60% sometimes. All right, so that's the neutrophils. They're the most common type. All right. The next most common type are lymphocytes. So you'll see a fair number of lymphocytes. Now, lymphocytes are the smallest of the white blood cells, so most of them are going to be about the size of a red blood cell. And you're going to see that they're mostly nucleus. They got big nucleus and a little rim of cytoplasm. Those are lymphocytes. They make up anywhere from 25 to 40% of the white cells. I think that's what I put on your list. All right. This is a monocyte. Monocytes are the largest of the white blood cells. And they make up anywhere from about 3 to 8% of the white cells. Now, the color staining of this one here is a little misgiving here, because in our slides, the cytoplasm of monocytes and lymphocytes tends to be a pale blue color. This is showing you sort of a lavender color. But in our slides, a classic, typical monocyte will have a blue staining cytoplasm. But these are the largest of the white cells. Their nucleus is typically shaped like a kidney, or a kidney bean, or sometimes like a fat horseshoe. All right? So these are 3 of the 5 types of white blood cells. Oh, and by the way, these are platelets. These are also platelets. Some of this. When you look at your slides, you're going to see little purple specks scattered all through. Tiny purple specks. Those are platelets. Platelets. Now here are all of the white blood cells. Let's go over the ones we've talked about. Neutrophils, and I don't know why they put the cytoplasm here as gray, because in our slides it's not gray. It tends to be a pale lavender color. Here's a monocyte. Remember I told you that the cytoplasm has a pale blue color? This is more typical of monocytes on our slides. Lymphocyte. Huge nucleus relative to the cytoplasm. You see this little rim of pale blue cytoplasm here? All right, so those are the 3 we talked about before. Here are the other 2. This is an eosinophil. Some people have trouble saying that word. Start slow. Eosinophil. 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 Pretty soon you can say it fast. All right. Now, its name tells you. What's the PHIL stand for? Love. What does it love? It loves the eosin, doesn't it? The eosin component of Wright's stain. So, its cytoplasmic granules pick up the eosin component of Wright's stain. You see these reddish-orange granules here? You only see those in eosinophils. The other thing is that their nucleus tends to be bilobed. In other words, you've got 2 parts and a little isthmus connecting them right here. They're not very common. But on our slides, they're not uncommon. But for most time, they take up about 2 to 4% of the total number of white cells in a sample. And lastly, this is a basophil. It loves the basic component, or the crystal violet of Wright's stain. You may not see one of these tonight, because they're so relatively rare in normal blood. They make up less than 1% of the white cells in a normal blood sample. But if you see one, and by the way, if you do, flag me down so I can check it out, because if it is really one, we want the others to see it too. We saw several in the early lab. A couple of them were kind of marginal, but anyway, this defines a basophil. You can see the dark nucleus in here, but you see these purplish granules in the cytoplasm here? This is what typifies a basophil. So in normal blood, you don't find many of them. Now, there's certain conditions that will increase their numbers in peripheral blood, but these are the 5 types. And this is what we want you to be looking at on these slides here. Let's go over how we're going to do this. The scopes are up here. There are also some scopes back there. 2 different manufacturers. You can use either one that you want to. If you use those back there, be sure that you know that they go in sideways, not front to back. If you put them in frontways, you can't close the door. Now, these microscopes, you use microscopes in A&P1 to look at the tissues and all. So you should be familiar with how to use a scope. Remember, they are delicate instruments, so you'll want to handle them delicately. But let's just go over how to look at a slide. Not necessarily a blood slide, but any slide. So you got your scope, you've got a prepared slide. You're going to mount your scope on the platform in the clips. All right? Now, what's the next thing you're going to do once it's plugged in and turned on and so forth?
Focus it.
All right, and how are you going to start focusing? What I'm getting at is what objective are you going to start with? You heard here on the video, you got to look at these blood slides on high power. Are you going to put the slide on the scope and flip around the high objective right away? No. Hope not. Because you may get lucky and be in focus that way, but most of the time you won't. It'll be extremely frustrating. So what do you always do? You start on scanning or low power and get in focus. First with a coarse adjustment knob, and then the fine focus if you need it. Then you go to the next higher power. So if you start on scanning, you go to low. Fine focus, go to high. Fine focus, and you're in business. All right? So you're going to want to look at them on high power. Now, when you're looking at them on high power, assuming that the oculars are 10x, what's the final magnification if you're using high power?
40. 400.
400 times, right? All right. Now, these scopes also have an oil immersion objective that magnifies 1,000 times. We don't use ever in this course the oil immersion lens. High power is the most we'll use. So be sure you're not flipping around the oil immersion lens, because if you do, you're not going to be able to see anything in it. All right? So high power is the most that we'll use. All right. Now, on these blood slides, you saw in the preparation of the slide, you got a drop on the slide, and you take another slide and fan it out, right? So where on this slide is the blood, are the blood cells thickest? At the beginning point, right? Where will they be the thinnest? At the other end, right? So if you're down here where they're thick, you're going to find that the red blood cells are often clumped together, and it's not going to be a pleasant viewing experience. Whereas if you go to the other extreme, way down here, it may be so sparse, you won't be able to see much. So where do you want to be? Somewhere in between. So what you do is you put your slide on the scope and just move it around till you find a nice place where the cells are not clumped, where they're in sufficient concentration, not so thinned out, you can't see much, and then just move back and forth, looking for these different types of cells. Now, so the takeaway tonight is to be able to identify, of course, the red blood cells, of course, the platelets, but be able to tell the difference between the white blood cells. Because we're going to want you to know this. We're going to ask you about it. Guaranteed. All right? Now there's going to be, in another lab later on, an opportunity to look at these slides again and look at some white cells. But if you don't know what you're looking for when you get to that point, you're going to be kind of like a lost ball in high weeds. All right, so to help you